I'm Rick Barron, the Executive Vice Chancellor for Health Affairs at MU and the Executive Director for the NextGen Precision Health Initiative. Good afternoon and welcome to today's NextGen Precision Health Discovery Series. It's my pleasure to introduce our speaker, Dr. Ron Mittler. Dr. Mittler is Professor in the Division of Plant Sciences and Technology, the MU College of Agriculture, Food, and Natural Resources, and he's a member of the Interdisciplinary Plant Group. Dr. Mittler came to MU in 2019, and just since then, he's published over 50 papers, obtained over $2.5 million in grants, and is a highly cited researcher. I asked Dr. Mittler to share how vital it is to conduct multidisciplinary research, the kind of research that crosses intellectual boundaries, by using the science in one field to inform another. His work in plant science has application to plant and human health. For example, his research includes the study of how oxygen is essential for life, but also has a dark side. All cells can do and produce toxic compounds called reactive oxygen species. These compounds can attack proteins, DNA, lipids, and other cellular components and induce a dangerous state called oxidative stress. The damage caused by reactive oxygen species is blamed for aging, cancer, and a number of other diseases, including neurodegenerative diseases, which I study. But it's also fueled a whole industry of antioxidants. So what should we believe about that we read in the newspapers and magazines? And with that, I'll turn it over to Dr. Mittler, and welcome, Ron, for highlighting you in the NextGen Discovery Series.
Thank you very much. Can everyone hear me? Okay, so thank you very much. I'm very happy to be here, and I will talk about oxygen and reactive oxygen and antioxidants, and I hope I will give you some answers at the end. So let's start. We're talking about oxygen, and oxygen is a molecule that we all like, and it's very nice and symmetric molecule. It's so symmetric that even the electrons at the uppermost orbitals of oxygen are in parallel spins. And this is actually, although a minor detail, it's very, very important because this makes oxygen stable, and this is preventing, I guess, us all from dying by spontaneous human combustion, because oxygen can be really reactive. And how do you make oxygen reactive? Well, there's a lot of reactions in the cell that can take oxygen and change the form of the electrons in oxygen. So they can flip this electron and move it into this orbital, or they can add an electron or two electrons and so forth. And these reactions, we still have oxygen. This is still oxygen, but now this oxygen is not prevented from interacting with any molecules in the cell, and it's called reactive oxygen because it can now react, and it can function as something that we call a free radical, okay? So very quickly and briefly, what is a free radical? A free radical is a molecule that's unstable, it's missing an electron, and it would like to attack another stable molecule in the cell, damage it, and steal the electron, and oxidize that molecule and cause damage. And antioxidant is a molecule that have an extra electron that can basically donate it to the free radical and make it stable again. Okay, so what are reactive oxygen species? They're partially reduced or activated form of oxygen. Okay, so there's still oxygen, but oxygen that we either added more energy to it and flipped the electrons, or we started adding one electron to it, or second electron, or even a third electron, and this happens in the presence of iron and called the Fenton reaction. And this is really important because it generates hydroxyl radical, which is extremely reactive and really dangerous. So reactive oxygen species can be toxic to cells. So they're not prevented from interacting. They don't have these parallel spins. They can react. And they're also always producing cells because we live in an oxygenic environment, right? We breathe oxygen, there's oxygen in the atmosphere and so forth. So because they can be toxic, because they're produced all the times, they really had a bad reputation. You know, back in the 80s, ROS were, termed the bad boys of biology, and people were really warned about the dangers of reactive oxygen species. So where are reactive oxygen species produced in cells? Well, the short answer is almost everywhere. In the mitochondria, you have many reactions and proteins that can take oxygen and add an electron to it. In the endoplasmatic reticulum, if you're a plant, in the chloroplast, you'll make a lot of reactive oxygen. In the cytosol, on the plasma membrane, outside the cells, and so forth. So there's a lot of different parts of the cell that make reactive oxygen all the time. As a matter of fact, I'm sitting here and in my body, there's probably millions of reactive oxygen species molecule produced all the time, okay? And the problem or the bad side of the reactive oxygen species is that they can cause damage. So they can attack DNA, they can cause mutations that can cause cancer, or they can cause basically cells to die eventually if they don't repair those mutations. They can attack proteins and inhibit enzymes, important enzymes, and cause problems in metabolic reactions. And they can attack membranes and cause the membranes to basically get destroyed. And membranes are really essential for the cell. So really, reactive oxygen species are dangerous, okay? They're reactive now. And if you have too much reactive oxygen species in a cell, of course, like I said, it will attack DNA, RNA, protein, and membranes, and it will cause or lead to a state we call oxidative stress. So if normal cells, maintain a normal balance of reactive oxygen, suddenly we have too much reactive oxygen, it will start undergoing cell death. It will die. And this state of oxidative stress occurs when you have really high levels of reactive oxygen so the cells are producing really a lot of it and they cannot detoxify. So I'll get to this detoxification in a minute. So really could be a problem. And in fact, because we're living in an oxygenic atmosphere, ROS and oxidative stress have been associated with almost all diseases that you can think of. I mean, that's a list of a bunch of different diseases and conditions that reactive oxygen and oxidative stress are associated with, okay? So you can find cancer, and you can find problems, heart problems, blood vessels, immune responses that are problems, and so forth. And that's the reason that back in the 80s, people were pushing, there was a big push to actually consume a lot of antioxidants. I even remember my parents sitting at the table having 20 vials of different antioxidants and taking them. So there was a big push to do that, but is it really required? So I hope I'll give you an answer at the end of this talk. Before I mentioned that cells can actually detoxify reactive oxygen species, and that's true. They have a lot of enzymes. So here are some examples. These are just examples. They have many more than that. Here's an enzyme called superoxide dismutase, and this enzyme can take superoxide, which is a ROS type of ROS, and make it into hydrogen peroxide, which is another type of ROS. But then enzymes such as catalase or peroxidase take this hydrogen peroxide and convert it back into water and oxygen. So you can get rid of superoxide and hydrogen peroxide. Cells also have antioxidants such as ascorbic acid, vitamin C, or glutathione. Some you need to consume. As humans, we need to eat ascorbic acid. Rats, by the way, don't have to, and plants make it all the time. And glutathione we make all the time in our bodies. So we have our bodies, our cells, and the plant cells have a lot of many different enzymes and antioxidants that are used to scavenge reactive oxygen or detoxify reactive oxygen species. As a matter of fact, there are many different pathways. I'm just showing this to just make the point. This is in a plant, but animal cells have the same many different pathways that evolved over millions and billions of years to protect any and every compartment of the cell, inside the nuclei, inside the mitochondria, in the cytosol. These pathways they use these enzymes and they use antioxidants, and they make sure that reactive oxygen species are kept at a low level. So just to give you an idea of how that happened, if you look at this is the scale of evolution of life on Earth, essentially billions of years, okay? We're here, right? So about 2.5 billion years ago, actually oxygen started appearing in our atmosphere. That is an event called the Great Oxidation Event. And that happened because earlier than that, cyanobacteria, which are unicellular bacteria, started doing photosynthesis that released oxygen. So about 2.5 billion years ago, oxygen started accumulating in our atmosphere, okay? And then before it even went higher than what it is now, and this is the level that we have now, okay? And that means that for at least 2.5 billion years, cells had to deal with reactive oxygen species somehow. And of course, there was no Walgreens or Walmart to go buy antioxidants. So they had to evolve all these mechanisms and all these enzymes and use antioxidants to protect themselves, okay? And we and others did some studies to look at how or when these enzymes, I mentioned superoxide dismutase and catalase, when these enzymes actually evolved. And this is, again, this is a scale. So origin of Earth is about more than 4.5 billion years ago. Origin of life is a little bit more than 4 billion years ago. And we found that about 3.5 billion years ago, they started evolving these enzymes, okay? And this is way even before the great oxidation event, okay? And what we need to remember that it doesn't really matter 2.5 or 3.5 billion years for evolution to be able to protect yourself from reactive oxygen species. And all this happens even before we had an organism that's made from more than one cell, a multicellular organism, okay? So this is, by the way, some biologists call this the boring years because there were only one type of organism, single cell organisms, many different types, but only single cells, before something really interesting of making more than one cell into an organism, way before dinosaurs and way before us, someplace here. So cells evolved many ROS toxifying enzymes and pathways over billions of years because they had to protect themselves from reactive oxygen species. And really what we're looking at this time and in our evolution is a balance, okay? So what happened is cells have a balance. There's a constant interplay between ROS production and ROS detoxification. So yes, there is a lot of ROS being produced in cells in my body right now, in your body right now, different metabolic reactions, different signaling reaction, immune responses, if you smoke or if you go and take a UV exposure bath, and many other problems and things in the body can make ROS, okay? So there's a constant production of ROS, but at the same time, there's a constant process of ROS scavenging or detoxification. We have enzymes, we have antioxidants, we keep iron level very, very low because otherwise we'll have a lot of Fenton reaction, which is bad. We shield a lot of the iron sulfur clusters in our body and so forth. So then we protect, so there's an interplay that makes sure that ROS doesn't go to really high levels and cause oxidative stress, okay? And that will cause cell death. So if you keep ROS at a low level, which then it can be regulated or can be used to regulate physiological responses, which I'll talk about in a minute, then you're actually in a good state. And that's achieved by balancing these processes. And in recent years, we actually realized that ROS can be moved from one part of the cell to another. So animal cells, plant cells, we have these channels in our membranes called aquaporins, and these channels can move water from one side of the membrane to another, but they can also move hydrogen peroxide. So if hydrogen peroxide accumulates to levels that are too high, it can be pushed outside the cells, or if it's needed, it can come from outside into the cell or move from the mitochondria outside. So it's not only scavenge, but you can also essentially transport, okay? So now we have a balance, it's a constant interplay between ROS production, detoxification, and transport, okay? So transport to another place, make it less toxic, detoxify it, and prevent oxidative stress, okay? So what are those physiological responses? What is the good side? Why do we need ROS, okay? So in the last 20 years, we've started realizing that ROS actually are really important for many, many reactions that are happening or many processes that happen in our body. So one of the first example was macrophages. Macrophages are white blood cells that trap bacteria, and then they bring them in and engulf them inside the macrophage into a vesicle called phagosome. This is the microbe, it's inside here. And then they make a lot of ROS inside that phagosome, and use some other compounds like chloride to make secondary ROS that kill the bacteria. Okay, so the ROS is being made only inside here together with antimicrobial proteins and other compounds and kill the bacteria. So it's important to kill bacteria that macrophages consume. ROS is also important, we found, for many different reactions in the cell, many signaling reactions, many pathways, regulate proliferation, regulate survival, regulate growth, regulate metabolism, angiogenesis, wound repair, many things. So a lot of these things need ROS to regulate them. Even stem cells, stem cells, if you don't give them any ROS, they will not differentiate. They will just stay where they are. And if we need them to repair or for other things, then nothing will happen. They will just stay there, self-renew, and not do anything. So stem cells need some ROS, okay, to start differentiating to what they need to differentiate to. Of course, if you give them too much ROS, then it will be really not good because they will undergo senescence and eventually die. ROS is also very important for signaling, for detection of stress. So if you take a plant and you give it stress, it will start making ROS. Okay, now ROS, of course, can kill the plant, but this ROS is used as a signal. The plant says, oops, I have too much ROS. It means I'm under stress. And then it will activate defense responses, acclimation responses. It will activate ROS scavenging mechanism. It will adjust the metabolism so the stress will not interfere so much. So maybe ROS are not that bad because we use them, our bodies use them, and maybe over evolution, actually we became to be dependent on ROS for a lot of the reactions and the mechanism and the processes that occur in our body. Another thing about ROS is that we now know about a number of examples of organisms that can survive extremely high levels of ROS, okay? So some bacteria in our mouth, some of those mouthwashes have high concentrations of hydrogen peroxide, and they can take it. They're okay with it. They're surviving it. Also, bacteria have been found in nuclear reactors, in the water of nuclear reactors, or some fungi and bacteria have been found in extremely high levels of radiation, like in Chernobyl. And high levels of radiation makes tons of ROS all the time, okay? And they still can take it. So again, maybe ROS are not that toxic, okay? So all these new findings are kind of changing the way we view ROS. And so maybe no more the bad boys of biology, maybe the darlings of biology, okay? So maybe they're actually required for life. Maybe we became dependent on them over evolution. And this kind of try to summarize this. So if this is the level of ROS, if the level of ROS in the cell is too low, the cells will go into a cytostatic state and they won't be able to do much. Of course, if it's too high, you'll go into oxidative stress and you can die. But if you keep ROS and what we call the redox biology range, and you do that by scavenging, production, transport, preventing iron from being free, and then the ROS can actually be used to regulate many different, proliferation, stress responses, many signaling reaction, differentiation, development, may regulate metabolism, defense, and so forth. So ROS can actually do their good functions if you keep them in the right range. And I think now we do understand that it requires 4 different things, okay? It requires scavenging, it requires transport, it requires chelation of iron and other toxic metals, and it requires repair. So if a lot of things in the cell make ROS, okay, you have ROS accumulating, you need to scavenge and transport them so to maintain the level, really normal low level, so you can use it for regulating protein structure and regulating signaling reactions. I'm not going to go into all this redox biology right now. But you also need to chelate iron, prevent hydroxyl radical from forming. And at the same time, you need to have an active process of repair. So any damage, you need to repair. So this, if you have all these 4 parameters working well in your body, you're fine. You're not in any danger of getting damage from reactive oxygen species. Of course, ROS can be good for you, but they still have a bad side, and we always need to remember that. And if those 4 components that I just mentioned, the scavenge, transport, repair, and chelate, if one of them even break down, then you are going to find out the bad side of ROS and you will be in trouble, okay? So today I want to briefly discuss 3 projects that are undergoing in my lab. So the first thing I want to talk about is balancing ROS and iron, basically trying to prevent this dreadful Fenton reaction that can make hydroxyl radicals. And we study proteins that do this in animal and plants. Then I want to talk about ROS as key signals for stress responses in plants. And at the end, I want to talk about some new findings that relate ROS to climate change and some problems that we might, you know, be exposed to in the near future. Before I do that, I want to tell you a little bit about my collaborations. I mean, this is an interdisciplinary, cross-disciplinary research that I'm doing. So, you know, in Mizzou, we have team science. We do science in different teams. People that I collaborate with are found at the NextGen. I collaborate with Camila Manrique, Diana Grant at the Bond Life Science Center, Ruti Angelovici, David Mendoza, Bing Yang, at the School of Medicine, Steven Segal and Joshi Trupti, Kafner, Felix Fritchi, and Heike Booking, and the IPG, Rick Ferrari and Manny Lishcombe. And of course, I have international collaboration in Israel, Belgium, the UK, and Poland. So let me start talking about the first subject, and that's balancing ROS and iron. And last 15 years, I've been working on this group of protein that I discovered in plants, but people have discovered it before in animals. I didn't know that until I found them in plants, and then I started working on them in animals. So I'm going to show only the data that we have from mammalian cells. So these proteins are called NEAT proteins or CISD. If you do cancer research, you know CISD more than NEAT. And in humans have 3 of these proteins, CISD1, 2, and 3. And the uniquely thing, they have iron-sulfur clusters that I'll talk about in a minute. So in these proteins, what we found are very important in regulate the levels of iron and ROS in mitochondria of mammalian cells and in chloroplast of plants. And in mammalians, it's really interesting because if you look at cancer cells, they have very high levels of NEAT proteins. If you look at beta cells or diabetes situation, you have low levels, and also these levels of protein declines during aging. So again, you need to balance them to have a healthy life. So we decided to work on cancer. Part of my position is at the surgery department here at the medical school, and I work on cancer because people at the surgery department, researchers, work on cancer. And this is studies that I'm going to show you have been done in collaboration with Professor Rachel Nechushtai from Israel. And what we did is we found out that cancer cells have a lot of these proteins. So we wanted to find out what happened if we suppress the amount of these proteins in cancer cells. And what we found is if you suppress CISD2 in particular, you suppress tumor growth very dramatically to 10%. So this is the example of tumors, and this is like the growth of the tumors in the animals that we do. All this research is done in mice using human tumors, basically. On the other hand, if we make the tumors make more of this protein, the tumors become much larger than normal tumors. And after we published some of these results, people are now using CISD2 as a diagnostic marker for several cancers. Okay, so that's really is really good correlation and prognostic and diagnostic of what will happen in certain cancer and how you should probably treat them. So we also wanted to see what is the role of this cluster, this iron-sulfur cluster. And what we did is that we overexpress this protein in cancer cells. And of course, you get much bigger tumors. You can see here the tumors are big. But now we overexpress this protein, but we did one amino acid mutation. And this is changing this histidine here to cysteine, and we stabilize this cluster. So now the same amount of protein as a normal CISD2 protein, but with one amino acid mutation, and look what happened. The tumors are really, really small. So what we think is that we need to target the cluster of these proteins to have a way to try to treat cancer. And that brings me into a translational science, meaning how to use science for really for medicine, and then that's a big part of NextGen. And we're looking at peptides. I have an NIH grant on this. We're looking at peptides that are derived from the CISD2 protein. And what we found, again, in collaboration with Professor Nafushtei, is that they can kill cancer cells, and they do it selectively. What do I mean by that? If I take normal cells and cancer cells, and I give them the same amount of peptide, and it's labeled with a green fluorescent label, you can see that very quickly the peptide bind to the membranes of the cancer cells, but nothing happening here in the normal cells. And eventually it goes into the cancer cells. And not only that it goes into, it kills the cancer cells, and it doesn't kill the normal cells, okay? And here you can see a movie of, these are normal cells, and these are cancer cells treated with the peptide, and you can see that the cancer cells are starting to shrink. You can see the green fluorescent protein of the peptide. And they will start making these blurbs, these bubbles here. You can see blurbing. This is a process, they're dying by a process called apoptosis. On the other hand, the normal cells are not doing anything. As a matter of fact, you see this cell is so happy it's dividing. While the cancer cells are dying, and you can see them all are going to start blebbing like that, the normal cells are doing fine. There is one stage after they divide that the peptide is incorporated into their membranes, but it doesn't kill. We also did this study in mice. So we took mice that has the human cancers, okay, and we gave them the peptide, or we gave them a very, very strong chemotherapy agent called doxorubicin. And doxorubicin is called the red devil. I mean, this is very strong. It's very potent. You cannot give it to people too many times because they will die just from the chemotherapy. And what you see here is that the peptide has a nice effect on shrinking the tumors, not as good as doxorubicin, but it doesn't have the negative effect. So if that's how the weight of the mice is dropping here because of doxorubicin, because they really look bad, and actually one of them died during this course of this therapy, the mice that get the peptide are happy. They're even heavier or gaining more weight than the normal mice. So in combination, perhaps with other treatments, that could be an interesting way to target cancer cells. Another thing that we're doing is we're looking at what happened to cells that don't have that protein altogether. And there is a rare genetic disorder called Wolfram syndrome type 2. And these are people that don't have CISD2. And they don't live past 25 years usually. They develop very early diabetes. They become blind, deaf, and so forth. And it's not a curable disease. And what we did is to study this, we developed a mice model for Wolfram syndrome type 2. And we looked at the effect of the deficiency of this gene on the heart. And I'm going to make a very long story short. What we found is if you take a young mice that doesn't have this gene, it's very similar to a very old mice in the fact that they both accumulate iron and they have disruptions in the mitochondria and ER, and they will probably eventually suffer heart failure because of that. Okay, so to summarize this part, need proteins or CISD proteins balance iron and ROS levels in the mitochondria of mammalian cells. I haven't shown you work in plant, but they do the same thing in the chloroplast. Of course, plants don't have cancer, so we're looking at other aspects of that. CISD2, this particular one, plays an important role in cancer and diabetes, and its level needs to be kept under tight control. And this is also important for keeping iron levels under control in hearts, and especially during aging. And then we think that we can propose a peptide-based anti-cancer strategy based on this neat protein function and based on the fact that the peptide is, derived from this protein, and we think we know what it does. But again, this is just a paper that was submitted. I don't want to discuss it any further on that aspect. So I will switch gears now and talk a little bit about plants. So that's about how ROS play a key role in the defense of plants against stress. Okay. And as you all know, the plants live in a very dynamic environment that can be really extreme. There can be really rapid changes, in light intensity, in temperature, in humidity, in insects coming and attacking it, sudden flooding, waterlogging, injury, air pollutions, and many more. So to survive, plants need to rapidly respond to these changes. They cannot basically move like an animal, go hide in a cave or something. They're standing where they are. So they need to respond very rapidly, and they need to be able to change their metabolism with a process we call acclimation, and be more resistant or resilient to the stress. And one of the more important processes that occur that allows them to do this is called systemic signaling. So if they sense any stress in any part of the plant, that part will send a signal that will go to all parts of the plant and alert all the plant that there is a stress, and it needs to be now starting to acclimate, meaning it needs to start synthesizing new proteins, accumulate new compounds, be ready, okay? So this process is called systemic signaling. It's the ability of a plant to mobilize a signal from basically a small group of cells to the whole plant. And this can be triggered by different abiotic stresses, pathogen, insects, and so forth. And once this systemic signal reaches the target, all the parts of the plant, it will activate different responses in these tissues and enable the plant to acclimate. So this is really important for acclimating to different stresses. Now remember, plants don't have a nervous system. They don't have those long cells that can send a signal from the tip of your hand to your brain in under a second, right? So they need to have a different solution to transducing or moving signals all over their size. I mean, some plants are really big, like trees, right? So you need to move this signal in a way that is efficient. So back about 10, 11 years ago, we discovered one of these mechanisms we termed the ROS Wave. And that's a mechanism that allows plants to send a signal from cell to cell. So there is no one long neuron, right? So you have to, if this cell senses the stress, it needs to tell the cell next to it, and then it tells the cell next to it, and so forth, right? And this is what we found. We found that if we give stress to this cell, it will start making reactive oxygen. And that will make the cell next to it make reactive oxygen. That will make the cell next to it make. So this is sort of a wave or a domino effect in which a signal can be propagated from this side of the plant all the way to this side. Okay, and then the rate of this, we have actually found up to 8 centimeters per minute, but mostly it's about 0.5 to 4 centimeters per minute, and that's not slow for a plant. And you can actually transverse a signal all the way from one part of the plant to another. So when I came to Mizzou in 2019, I purchased this imager, and this imager is a fancy imager used to image mice. But as I told you, I'm doing research in plants and mice. So I started asking the question: can we use that to image ROS in a whole plant? And we developed, together with my postdoc, Yosef Fitchman, we developed a system to actually image reactive oxygen. So what you see here is 2 plants. Okay, so one was never treated, the other one was treated only on this leaf. And then you can see that over time it accumulates reactive oxygen species in all the plant, although we treated only this leaf. Okay, and you can do wounding, you can do highlight, you can do pathogen. And here's a movie showing this. So this plant was treated only in this leaf, and this plant next to it was not treated. And you can see in real time accumulation of reactive oxygen in live imaging in a plant growing in soil. That has not been done before, as far as I know. And that allowed us to basically do a lot of many different things with plants and study this process of the ROS wave. That was the first time it was actually visualized like this. So we made a movie about this. I'll let it run right now.
Plant cells respond to stress by sending signals from the tissues that first sense it to the entire plant. This type of response is called systemic signaling. The systemic signal moves from cell to cell over long distances at rates of up to several centimeters per minute. But how does the signal propagate? Changes in calcium levels in the first cell trigger the production of ROS at the apoplast by enzymes called RBOs. Triggering more ROS production and opening plasmodesmata, allowing the signal to move from cell to cell. Once the signal reaches the next cell in the path, the process is repeated. And ROS production in the next cell is turned on. This process is called the ROS wave, and it is essential for systemic signaling. As a result, all plant tissues, even ones that did not initially sense the stress, become tolerant.
So another thing that we can actually measure is redox. So using this machine, you can see here, this is a picture of the same plant, okay? But here you can see the oxidation of the proteins that occur, and this is showing you the reduction of the protein. So when you treat this leaf here, it's the same leaf, right? You can see that there is more oxidation occurring in the plant because ROS is reaching all these part and oxidizing proteins there. And this is actually important because this allows the ROS to change the redox state of the cell and trigger many different defense mechanisms, okay? So again, just show you this movie again. So this is the same plant, okay? This leaf is treated. This is showing only reduced protein. This is showing oxidized proteins. And you can see that the intensity here decreases while the intensity here increases, which means that the whole plant is undergoing a change in the redox state, which is very important because that will trigger many different defense mechanisms, okay? And here's another neat experiment. We took plants and we gave them two different stresses, injury and highlight, on two different leaves, or on the same leaf, or separately. And you can see that the ones that got the two different stresses on the plant that got them on two different leaves makes a ROS wave that is much stronger than the plant that got them on the same leaf. Okay, and it's even stronger than just highlight or injury. So plants can actually integrate different signals that come from ROS production and become more tolerant to stresses. So there's a lot of work I'm not showing you. Essentially, if this is the ROS wave, this is us trying to identify the molecular mechanism underlying the ROS wave and how is this progressing. You know, occasionally we can get some oxygen from the granting agencies and we can keep swimming under the waves and trying to figure out what's happening. So in summary, the ROS play an important role in cell-to-cell communication in plants. We even believe that that originated early in evolution, and we're doing some studies on that. And I showed you that a wave of ROS production is progressing from any kind of signal, a wounding, highlight, a pathogen. And this I haven't shown you, but it triggers a lot of defense mechanism and makes the whole plant resistant. This wave of ROS drives a redox wave that can activate defense and acclimation pathways. Plants can integrate two different systemic ROS signals. And these signals, these systemic ROS signals are primarily mediated, I haven't shown you that, through the vascular bundles of plants and require these ARBO or NOx type enzymes. So last part I want to talk about is multifactorial stress combination. And this is something really that I've developed over the years. Ever since I studied desert ecosystems back when I was in Israel. And I realized that, you know, if you go, this is a picture of a desert ecosystem where I used to do my research. In the lab, more than 99% of scientists usually test one stress condition at a time. So they will only do heat stress, or they only do drought stress or cold stress. But in nature, organisms are always simultaneously exposed to multiple stress conditions. So in this ecosystem, there was low nutrients in the soil, there was high salinity in the soil, there was low water, there was hot in the day, cold at night. So it's not, you cannot just say, oh, I know one thing and that will apply to everything. So I've been studying for many years now drought and heat combination. And the reason it's important is shown in this slide. This is what you summarized from 1980 to 2012. If you take all the weather disasters that exceeded $1 billion each, okay, and you put them together, you don't put hurricanes and tornadoes here, and this is droughts, okay? This is freezing, this is flooding, and this is drought with a heat wave, okay? So you can see that when drought is combined with a heat wave, it's much, much worse than drought, okay? So this is why we're interested in that. But in the recent years, I've realized that it's actually much more serious than that, okay? So what happens on Earth right now? So we are humans, right? We're growing in our population, and at the same time, we're using more and more agriculture, prime agricultural land, converting it into cities and towns and so forth. So the amount of land is decreasing. Because our population is increasing, okay, we're using more fresh water. Our global freshwater use is increasing, and the renewable freshwater resources are decreasing, okay? Now, at the same time, we're also producing a lot of greenhouse gases and causing global warming. So this is the CO2, atmospheric CO2. This is global temperature anomaly. Our planet is warming up all the time. That process, global warming, is causing climate change. And our climate is changing to the worse, actually, because we get more droughts, more heat waves, more cold snaps, more flooding, more storms. This is a graph that shows from 1960 to 2019, what happened in the frequency of droughts and events of extreme temperature that many times happen together. Not always, but many times. But they're increasing, the frequency is increasing. And the intensity is increasing sometimes too. Now, on top of that, we generate a lot of pollutants, right? So microplastics, we use pesticides, we use burn fuel consumption, and a lot of many other things, okay? So it's not only two stresses, okay? Now we're talking about a multifactorial stress, okay? So low water, storms, droughts, heat waves sometimes, global warming, day-night temperatures are increasing and so forth. So how is that going to affect plants, animals, and microbiomes? You know, just sent a grant about this to NSF. This is shown here in this principle, kind of trying to introduce this new principle in plant biology, okay, new concept. It's not only one or two stresses. We're talking about, you know, a plant can grow in soil that has pesticides or herbicides or microplastics, or residues of diesel. There's persistent organic compounds, heavy metals. There's a lot of pathogens now. There's the climate threats I talked about. There's other problems with nutrients, salinity, pH in the soil. I'm not saying it's all affecting at the same time, but different parts of our globe are affected or subjected differently. So in some seasons, like the Midwest, will have flooding and heat. I'm not talking about all the contaminants, the other contaminants. The West has been suffering for many, many years now from drought and heat, right? In Australia, they're really lucky. They have drought, heat, and high salinity. So add to this all the other factors and you have a problem, okay? And it's not getting better. So we decided to check this, recently published this paper about what happened to a plant when you start subjecting it to multifactorial stresses. And we used heat, salt, highlight, paraquat, which is a herbicide, acidity, soil acidity, and cadmium, which is a heavy metal. And what you can see here is survival of plants, okay? And we use wild-type plants and two mutants that are deficient in ROS scavenging and production. So these are deficiency in ROS metabolism, okay? And what you can see is that if you apply only one stress, you almost cause nothing. We use specifically low levels of each stress. So one stress by itself almost had no effect on survival. If you start to do all the different combinations of two stresses, you're starting to have an effect. But the real problem is showing up at 3 and 4 combinations. And remember, each of these stresses by itself had no effect almost, okay? And if you start having more than 3 or 4, then you're basically having a bad effect on the plant. There's 20% survival, and the mutants that are deficient in ROS metabolism and signaling are even worse. Okay, so this brought us to this kind of new principle. We call it the plant multifactorial stress principle, and I will read it out. So with the increase in the number and complexity of stresses or stressors impacting a plant survival will dramatically decline. So this is the number of stresses simultaneously impacting a plant, and this is survival. And this is even if the level of each individual stress involved in this combination is low enough to not have a significant effect. So one stress has no effect, okay? But once you start piling them up, compiling them up, you have very bad effect. And this, I think, it's a really problem because, you know, if you go out and you look at the field and you say, well, you know, yeah, there's a little bit of drought and a little bit of salt, but it's fine. Yeah, but keep adding a little bit of this and a little bit of that, and eventually your plants will die. A similar principle has been shown now for microbiomes by Relig in Germany, and the same principle. So if you start piling different stresses, the microbiomes will decline. So the ecosystems can also decline. So we're in a tough situation that we need to be really careful, I think. We've also looked at many different mutants and how they respond. And interestingly, if you knock out a NEAT protein in the plants don't survive this multifactorial stress. If you overexpress the NEAT protein, they have a better protection a little bit. So what might be wrong for mammalian cells, because you don't want to have too much NEAT in mammalian cells because you have cancer, will not be very good for plants, actually. So that's another way of maybe thinking about, you know, cross-thinking about how to handle different, you know, stresses or different conditions using knowledge that obtained from different fields. So in summary, multifactorial stress combination occurs when multiple abiotic and or biotic stress factors are impacting the plants simultaneously. Global warming and climate change and industrial pollution increase the frequency, complexity, and intensity of these combinations. And with the increased number of factors affecting the plants, the survival and growth will decline, even if the level of each stress is very low. So this might be we might be blind to this effect, but we'll find it out later. And then this harmful effect of multifactorial stress combination should serve as a dire warning to our society and prompt us to drastically act to reduce the different sources of multifactorial stress in our environment. Okay, so I want to come back to these questions. Are antioxidants the solution? You know, I told you that ROS and oxidative stress have been associated with almost all diseases that we know. And in some cases, rightfully, because if this chain of scavenge, chelate, transport, and repair is broken, you can have problems that are associated with any of these diseases, okay? So the answer is, and I'm not giving you the answer, I'm giving you an answer. And the answer is that based on your age, your genetic makeup, your own genome, because we each have a different genome, right? Your level of activity, your health condition or conditions, your environment, your stress levels. Based on all these factors, you need to balance the intake of antioxidant. And if possible, use natural resources. I always prefer to have the natural resources over these chemically produced antioxidants, okay? And you have to remember, ROS are both good and bad, so you need to have a balance. And I think this is where precision medicine or NextGen come into play, because it's personalized, okay? It's like personalized medicine. So you're different. People are different from each other, so they will need a different intake. They have different activities. Athletes might need more than couch potatoes, right? So it needs to be tailored. And I think that in the future, you know, there's going to be an app for this, because you'll have your genome sequence, highlights of this sequence will be incorporated into this app. Probably it will be sitting on your wrist, taking information about you all the time, and it will tell you, hey, Ron, this morning you better take that amount of vitamin C or something like that. So this is where we need to be, you know, advancing forward, I think, in personalized medicine, and that's something that NextGen is here to help with. So we have a joke in chemistry that all chemists never die. They just reach equilibrium. So I think that if you reach an equilibrium between the levels of antioxidants in your body and the levels of ROS, and you stay safe in this room from any accidents, you can live to the full potential of your genome. Okay, of course, if you get out and do extreme sports and put yourself in danger and smoke, you may not live to the full potential of your genome. So that's something to keep in mind. Balance. Okay, so I want to thank a lot of the people. Nobuhiro and Gadi were the first people, postdocs that work on the ROS wave. Yosef developed this new imaging method, and together with Sarah and Ronnie and Amit made a lot of discoveries about the ROS wave. Lua and Linda and Ola worked a lot about the NEAT proteins. Sarah and Ranjita and Maria are working on stress combination. My research is funded by NSF and NIH. There's a partial list of more collaborators, and some of them were repeated before. After I've shown you how my people look, I want to show you how my people see me. So this is me in the lab. I want data, feed me data. And they know, I'm hoping they're there watching and laughing. So with that, I will conclude. Thank you very much. I'm happy to answer any questions.
Ron, that was great. Thanks so much for that talk. And so we can really get an understanding of ROSs. In physician clinics, ROS is review of systems, but here it's reactive oxygen species. So that was great. So I put a chat in. It's hard to think of examples where there are actual proven therapies in humans of drugs that decrease reactive oxygen species, but one is in my field. In ALS, 3 years ago, edaravone was improved. It's an IV infusion that ALS patients have to take 14 days out of the month. And the mechanism is reducing reactive oxygen species, and it seems to slow down the disease by 30%. It's approved in Japan for stroke, but it never got approved in the US for stroke, but it did for ALS. There's not too many examples of like that in medicine, which have shown that, but that is one. It's a big one.
Yeah, I mean, I can give you a few others. For example, in transplants, when you transplant an organ into a patient, that organ has been sitting on ice usually, and it has very low levels of oxygen. And when you hook it to the bloodstream, a burst of oxygen can come into this new organ and actually cause oxidative stress. So they add to the person's blood some mimics of antioxidants to prevent that. So these are small compounds like SOD mimics and so forth that will help prevent ROS from attacking or right. the onset of opening the blood into the transplanted organ. So that's another example.
Thanks, Ron. Mary, go ahead and read some of the other questions for Dr. Mittler.
Great, thank you. Dr. Mittler, the first question starts out with great presentation. And how does the ROS wave look like for a pathogen such as a virus which spreads systemically too? And second part, and what pathogens and what do pathogens do in order to circumvent the ROS-mediated defense response?
Okay, so we have not tried to look at viruses. I would think that the ROS wave, I know that the ROS wave is faster than the spread of a virus through the plant. So if there's a wound response, if there's a wound, that usually viruses enter a plant through a wound that can be from a pathogen, from an insect eating the plant, or from like mechanical injury, and that will trigger the ROS wave. And then the spread of the virus is really slow inside the plant compared to the rate of the ROS wave. So the ROS wave might have a function, but we've never tried that. We looked at different bacterial, or we look at bacterial pathogens, but there's so many other pathogens. There's actually some smart fungi that trigger the plant to make ROS and then use that ROS to kill the plant and eat the dead tissue. So this is biology. Biology is amazing in the ways that things happen and organisms evolve, to take advantage. So some organisms will take advantage of the bad side of ROS further on to kill the plant and eat it. So there's a lot of examples.
Thank you. This next question, what are your top 3 to 5 sources of antioxidants?
There's a, I'm hesitant to say because, you know, fruits and vegetables are my top choices, but if they sit on the shelf for too long or they're in, you know, in storage for too long, the level of antioxidant decreases. So I would say fresh fruits and vegetables of almost any kind, you know, is a good choice.
Great. Thank you. And I'm hoping others continue to put in some questions here in the Q&A function. And this last question I have here, are we aware of what happened in 1980 that caused that weather event on your global warming slide?
Let me think. Oh, that was, I think there was a big heat wave and drought that occurred simultaneously. And that was really, really bad for agriculture. It caught agriculture basically unprepared. After that, the target of breeding, so egg biotech companies, big breeding companies are trying to, for years after that, have tried to breed crops that are more resilient to drought and heat stress. There was some success at the vegetative growth, but the challenge is really the reproductive parts. What we eat, for example, in soybean or in corn is the seeds, right? So if there's no reproduction, there's no seed production, we don't have the yield that we want. Yes, we will have, you know, a lot of plant material, but no seeds. And apparently one of my research projects is on how the stress combination is affecting the reproductive processes, meaning the fertilization in the flower, the seed growth itself, the embryo, and these are really, really sensitive to heat, and they're sensitive to drought combined with heat, and we're discovering new mechanisms that plants use to try to fight that and keep the flowers cool, even under conditions of drought and heat. So I can tell you that, for example, they will sacrifice the leaves just to keep enough water to cool down the flowers and give us some seeds so they can go on to the next generation. For us, it's food, but for them, it's the next generation and survival, basically.
Thank you. This next question, how does the peptide get into cancer cells?
Oh, that's the whole subject of the grant. So this grant is a collaboration between my group and a group in UT Austin, led by Ron Elber. And he is a mathematician, and he models membranes. And what we found is that the reason, so he modeled cancer membranes and normal cell membranes. And we found that there's a difference in the charge on the membrane of the cancer cells. And that probably is, he actually modeled the whole pathway. And then we did experiments, that's a paper in submission now, to prove that the model that he did is well, we checked it and it proved to be correct. That is correct. So the peptide binds the cancer cell membranes primarily because the cancer cell membranes are more negatively charged than the normal cell membranes. And then it enters them in a mechanism that is helped by the composition of the cancer cell membranes. It's not the simple answer, but it happens. I mean, we see it happen, so he can model it and give us a suggestion that we're now testing more and more.
Well, good luck on that submission. And with that, I'll turn it over to Dr. Barron.
All right. Well, thanks again, Dr. Ron Mittler. What a great talk. So once again, these webinars are really intended to highlight critical precision health topics and approaches. And we would like each of you to think about research opportunities in precision health where you can contribute to help identify potential collaborations across the University of Missouri system, like Dr. Mittler has done. Please join us again next month for our NextGen Discovery Series, where Dr. Melissa Terpstra will present Growing Team Science, Building Biochemistry into the Human Connectome Project. Dr. Terpstra is the director of the NextGen Imaging Facility. She's a PhD in medical physics and is an expert in uncovering very tiny signals in magnetic resonance spectroscopy using ultra high field magnet resonance imaging. Her presentation will start on Tuesday, April 19th at noon. And take care, everyone, and we'll see you next month. Thanks again for tuning in.
Thank you very much.